Okay, I think we're going to make a start. Welcome to the IICD's monthly seminar. Today's sort of fun for me because I'm about to introduce somebody I've known for a long time when he was a PhD student in University College London. And he visited me and Daryl Shibata in California and started learning how to do experimental work. So just to tell you what happened, Daryl, after Trevor's gone home, says, amazing having mathematicians in a lab. They make mistakes I've never seen before. Anyway, since then we've kept in touch, and Trevor has spent 8 years at Queen Mary University of London and the Barts Hospital, and then a year ago moved to the Institute for Cancer Research, where, let me get the title right, he's the head of the Center for Evolution and Cancer at the ICR, and he's made a lot of contributions to trying to understand the development of tumors. And for this work, he was fantastically elected to the Academy of Medical Sciences last year. That's the English equivalent of the National Academy of Medicine. So he's going to tell us today about some of these adventures, and then tomorrow he's doing more of it, perhaps different. Anyway, today it is inferring the drivers of cancer evolution using mathematics and multi-omics. So welcome.
Thanks, Simon.
Proceed.
It's lovely to be here. Thanks for the invitation. And it's nice to spend a few days here. As we were talking about over lunch with some people in your centre, that my lab skills haven't really improved since Daryl's days. So I focus more on the math side. But anyway, I tried. Yes, it's really nice to be here. As Simon said, I'm at the ICR in London, and I lead the Centre for Evolution and Cancer. And our centre is both a hub for computational biology within the Institute. A number of the labs and also the core informatics services exist within the centre. But we're also a big wet lab. And so we have lots of facilities for doing fancy measurements of cancer, including single cell sequencing and spatially resolved data generation and those kinds of things. So we're this kind of interdisciplinary environment with both experiment and theory. And hopefully my talk reflects that focus in the centre. And so really grateful for funding, particularly from Cancer Research UK and from the Wellcome Trust, and also a little bit from the National Institute of Health here, in a collaboration that's ongoing with Daryl Shibata ever since I spent some time with him in LA more than 15 years ago. All right, so here we go. So I want to talk to you about how we're trying to infer cancer evolutionary dynamics. So here's a depiction of what I'm talking about, and this will be very familiar to you all, I'm sure. So this is a drawing, but it's supposed to be a graph, and on the x-axis we have time, and on the y-axis we have the size of a clonal expansion within the body. And the blue patch is supposed to be healthy cells, and we think, of course, that cancer develops because there's some heritable alteration acquired by those healthy cells, probably a genetic mutation, but might be a heritable epigenetic change, which alters the phenotype of the cells, gives them an advantage, and they clonally expand. And that's that sweeping yellow wedge there. And then the red cross is supposed to indicate the acquisition of that heritable mutation or epigenetic change. And then that gives us a benign tumor, like the many moles on my body, and I get more of them as I get older. But that's not a cancer. And then progression to an invasive cancer or metastatic cancer, or even a cancer resistant to treatment, is through the acquisition of further important alterations, which might also drive a selective advantage. That's those sweeps of other colors, eventually resulting in that nasty cancer, the dark red on the far side of the plot. And so fundamentally, our work has been to try and work out these dynamics, to know what they are, and importantly, to know which mutations or which heritable alterations actually drive cancer evolution. And so another way of putting it is, we spend a lot of time looking for Ryan Gosling in the cancer genome. And that's really what I want to talk to you about today. How can we work out which mutations or other changes are Ryan Gosling? I don't actually like Ryan Gosling that much, but I thought it was quite amusing. All right, so what drives cancer evolution? And that's the goal of the work I want to describe today. So as you all know, the state of the art is using statistical genetics and doing it across very large sets of data. And so one of the very best pipelines out there is from Nuria Lopez in Barcelona, and it's called the Intergen pipeline. And the Intergen pipeline works like this. So you take as much data as you can possibly get, ideally thousands and thousands of tumors of a particular type. And you have sequencing data from each of those tumors. And then you assess each gene in turn, and you look at the pattern of mutations in that gene across all of the tumors in the cohort. And what Nuria and others have realized is that the pattern of mutations in a gene is different if that gene is under selection, if it's driving cancer evolution, compared to if it's just a passenger. So if the mutations accrue at random, they have a different pattern in the gene than if that gene is under selection. And so the patterns, broadly speaking, are these three. The first one is that you'll tend to get more mutations which change the bases in the, change the amino acids in the gene, more non-synonymous mutations than you would expect by chance if that gene is under selection. And that's the basis of methods like DNDS that I'm sure are very familiar to you. Also, the mutations will tend to cluster in functional regions of the gene. So a very classic example is the KRAS gene, where essentially all oncogenic mutations are in just 2 codons, because they're the ones that control the activation of the gene. And of course, if you have other functional information about what those individual mutations do from orthogonal sources, say in other diseases, not cancer, you would expect that cancer-associated mutations would be enriched for mutations which are known to be functional. And then so you can integrate essentially over these 3 different properties of those mutations and select out the set of genes which are enriched for patterns associated with selection. So that's great, and this works really quite well. And so if you, I'm sure you've already done it, most of you, but if you go to the Intergen pipeline and you say, tell me what are the likely driver genes in colorectal cancer, you get this list. And so this is a bar chart, genes on the X, and the number of samples with that mutation on the Y. And if you read the gene names, hopefully you can, you'll see that there's lots of familiar genes there, with APC and TP53 being the most frequently altered driver genes in colorectal cancer. So this is good, but let's just think for a moment about what this is. So this is telling us that on average, across those cohorts that are used in the discovery of these genes, that these genes are drivers. But what it's not telling us necessarily is that in an individual cancer, that these genes are driving that individual cancer. We know on average, most of the time, they will be driving a cancer when they're mutated of that particular type, but not necessarily in the individual cancer in front of us. And so this isn't just a detail. I think this becomes very, very important. So one reason it becomes very important is that every patient's evolutionary history of their tumor is different. So in a project ongoing in the lab, which I'm not going to talk about today, but it's, I think, an interesting one, we've been collecting samples over time from patients who have a primary colorectal cancer, which is then metastatic, so it becomes stage 4. And these are groups of patients who remarkably have stage 4 disease. The disease spreads all around their body. You can see the depiction here of the time course for this individual patient. But despite having this rampant stage 4 disease, they live a very long time, in some cases more than 10 years. And so we call them exceptional survivors. And they're clearly very different to perhaps a more average pattern that happens for metastatic colorectal cancer patients who live maybe just a few years after their initial diagnosis of stage 4 disease. So there's something clearly very different here. And what we found is these patients tend to have the same driver alterations as other patients. So you can see that genes are acting differently in different people. Another reason that identifying drivers on a case-by-case basis is really important is because we've been looking essentially harder and harder to find those mutations. So the next slide has a slightly gory image. If anyone's squeamish, then please look away for a moment. So what we've been doing, as many of you have been doing as well, and many people around the world are doing, is taking big ugly cancers like this. This again is a colorectal cancer, and we're doing multi-region sequencing on it. This is some work we published a few years ago where we took a relatively large number of biopsies from a single cancer and did whole genome sequencing on each of them. And then we can construct these phylogenetic trees. But this means now that we've sequenced lots more cells. And so, of course, because we sequence far more cells, we find more stuff. And if mutations just occur at some background rate, then if we look harder, we'll find more. And so it becomes challenging to know, well, what really is driving these cancers, because we find more and more if we sequence more and more. And so this is a good example of that. These are two other colorectal cancers that we sequenced more recently with multi-region whole genome sequencing. And here we've annotated the driver genes, or at least the mutations in the genes on that list of on average drivers that was identified by Intergen onto our phylogenetic trees. And you can see that in both cases, there's evidence of mutations in these likely important genes throughout that phylogenetic tree. And so nobody would question that these APC mutations, which are kind of a hallmark really of colorectal cancer, are important. But what about this PI3 kinase mutation that occurs later on, or some of these other mutations that are in subsets of the tree? Are they really drivers or not? They certainly are on average, but in this particular cancer, are they really shaping the evolution of this tumor? And that's the question that we want to try and address. So which are the real drivers in each case? Who's Ryan Gosling? So of course, if we could have this picture in real life of the time course of cancer evolution, this would be easy, because all we need to do is watch the cancer and say, hey, that mutation showed up here and it's associated with the clonal expansion. So bingo, that's a bona fide driver. But of course, we can't get longitudinal measurement. And so again, the next slide is a bit gory, so if anyone's squeamish, look away. The problem is that we can only sample at the end. So we only get to look at the big ugly cancer in our lab after it's grown. With a few rare exceptions, the only thing you get to sequence effectively is the end point of the process. You never get to directly observe the temporal dynamics of cancer growth. And so our approach, much like the approach of many of you here and many other people too, is to try and characterize the pattern of intra-tumor heterogeneity of mutations particularly, but also some other properties of cancer cells that I'll talk about at the end of the talk, and use those as a kind of readout of how the cancer grows. And so what we've realized, and others have realized too, is that the particular pattern of tumor heterogeneity is a direct consequence of how that tumor has evolved. And so if the cancer has evolved to have a subclonal driver alteration that actually makes some cells grow more than others, that will shape the pattern of tumor heterogeneity differently to if that driver wasn't there at all. And so the analogy I like is this rings in a tree. Like, we can think of the pattern of mutations across a cancer as the secret diary recording how the cancer grew. And of course, you can think of the rings in the tree as a secret diary recording how the tree grew. You can see the growth in each year and learn something by sampling from the rings about the environment that tree was in that particular year. So our goal is to try and read that secret diary of intra-tumor heterogeneity. And so we do it with an approach directly inspired by Simon that I might talk more about tomorrow, by combining maths with data and fitting them together using ABC. So what we do is we make mathematical models. That cartoon on the left is representing a stochastic branching process model, where we start with the first cancer cell. It divides at some point in time to make more cancer cells, and then they divide at some later point in time to make even more cancer cells, and so on and so on and so on. And we can simulate the acquisition of mutations as that virtual tumor grows. And then we can make predictions about what the pattern of tumor heterogeneity would look like. Essentially, what's the pattern of mutations at this last point in time that we simulate, given how we decided that tumor grows. So we have some parameters that describe the pattern of tumor evolution, and we can obviously adjust the parameters, and they change the pattern of tumor heterogeneity that the model produces. And then we can compare those predictions of our stochastic models to actual data. And so this is a depiction of whole genome sequencing data, and I'm sure representing the data this way is also familiar to lots of you. But here on the x-axis, we've got the allelic frequency of the mutations, the variant allele frequency. Instead, you could plot the cancer cell fraction if you normalize for copy number and purity. And then on the y-axis is a histogram, so we're plotting the number of mutations at that particular frequency. And so this data is a direct measurement of intratumor heterogeneity. The mutations at high frequency are the ones which are clonal, so they're not measuring tumor heterogeneity. The spread, of course, is because of the sampling of next generation sequencing from sequencing a small number of the total alleles in the tumor and the sampling that also happens on the Illumina flow cell. So, there should be a peak of mutations at some high frequency representing the purity and ploidy of the tumor, but it spreads out because of that sampling. So we don't really care about those for learning about the evolutionary history within the tumor. But what we do care about is these ones, which are at lower frequency. So they're subclonal, these kind of funny squiggles in my diagram. So they're in some, but not all of the cells in the tumor. And it's this allele frequency distribution of these subclonal mutations that is our secret diary. This is our readout of how that cancer grew. And so essentially we make mathematical models to predict what this distribution should look like. And then we change the parameters of our mathematical model to make it fit this distribution. And in that way, try and infer how that cancer grew. And so I imagine you can already see how we can use this framework to assess on a tumor by tumor basis the consequence of drivers, but I'll obviously try and walk you through it. Just as a note, if people aren't familiar with this data, this lack of mutations at very low frequency is false. This is just because we haven't sequenced deep enough. If we spend more money, I'm sure we'd find more mutations down here. It's just the limit of sensitivity of the machine. So of course, when we do simulation, we can also simulate those kinds of noise or technical error in our measurement as well. All right, so we've worked on this for a number of years, and you may have read some of the papers, so I won't go through the whole story because you'd fall asleep, I'm sure, or I might with my jet lag, but I'll just give you the summary. So the first thing we did a number of years ago was to ask, what's the most boring kind of cancer evolution you can have? And we decided that was neutral evolution, which is essentially when nothing interesting happens. It's when all the cells do exactly the same thing. So they all have the same probability of producing surviving offspring. So we end up with an exponentially growing tumor. And you can write down, if you solve the stochastic process here, that the frequency distribution of mutations for subclonal mutations follows a characteristic 1/f² power law. It's 1/f in the cumulative. And we can think of this as a kind of null distribution. So we can say if there's no interesting evolution going on, if it's all neutral, then our expectation about what the pattern of tumor heterogeneity should look like should be this 1/f² distribution. There's ever more mutations at ever lower frequency. And then we can also do simulations where we allow positive selection to happen. So rather than the tumor evolving neutrally, it evolves neutrally up to a point where one cell here flips red in my cartoon, and it flips red because it's acquired an advantageous driver alteration. Might be a mutation, but the model is of course agnostic to what the mutation actually is. It might be an epigenetic change. It could be that that cell is in a particularly advantageous microenvironment. But the net result is that that cell then produces more offspring compared to other cells in the tumor, so it clonally expands. And then we can ask, well, how does that pattern the sequencing data? And so what it does, it causes an excess of mutations at higher frequency than you'd expect compared to the neutral model. So, here you get this very clear bump in the distribution. This bump here is all the mutations that are acquired along this lineage in the cancer prior to this driver alteration being acquired. And then those mutations along this lineage here are dragged to higher frequency as that subclone expands. So this bump, as the subclone is expanding, moves from left to right in the frequency distribution. And in fact, you can write down what the mathematical properties of this distribution should look like as a function of how the cancer is evolving. And so the number of mutations in this bump, this peak in the distribution, is a function of the time elapsed since the beginning of cancer growth before that subclone arose. So if the subclone arises very early in tumor growth, there's not much time for the subclone to acquire new mutations, so the bump is very small. Whereas if it arises late in tumor evolution, it has lots of time to acquire new mutations, so it's a very large, pronounced bump. And then where the bump is on the x-axis, its frequency is proportional then to how fast it's growing compared to the rest of the tumor population. Because if it's growing very fast, it'll be at high frequency. And so if you know the mutation rate, then you can kind of make a molecular clock argument to say, well, the number of mutations is proportional to its age, and therefore I know when it showed up, and I know how big it is now, so I can work out how fast it's growing. So from these data, just from this snapshot data, with this underlying mathematical model, you can start to get dynamics about what's happening in these cancers. I think this is clear to everyone, but just to labor the point, here you see selection, you see that there's a driver alteration there, not because you found the driver, but because that driver causes all the passenger mutations to be overrepresented. And it's the passenger mutations that give you the signal of the driver being there, not the driver itself. So it's a totally distinct approach to that intergen pipeline I described to start with, which is trying to find the drivers directly. Here, we're just looking at the consequence of there being a driver on the rest of the genome. And so we get a kind of orthogonal way to go at the problem. Yeah. Sorry.
How do you spot it? It could possibly also just like a driver is mutating, like the gene that is driver has like more mutations and getting more.
Yeah. So, yeah, sure. Yeah, that's true. So the question was, how do you know the alternate hypothesis, which is selection, I guess, in your framing, is true? And of course, we never know anything for certain, but what we can do here is a kind of Bayesian model selection argument where we can say, what's the most likely explanation for this pattern in the data? And we're not certain, of course, that's the explanation, but we can give it a probability and ask, when is that more likely than the neutral case? Because you do sometimes, which I think was implicit in your question, see this pattern of what looks like selection in a neutrally evolving tumor in our simulations, just because you've got neutral drift and you just caught it at the right moment in time. So yeah, you can never be definitive, but you can say what's most likely or has the highest posterior probability, to be precise. Okay. So you can imagine an approach now, I think, where if we've got a cancer, we can sequence it and we can say, this cancer has selection going on most likely because the data looks like this. And then is there a driver in this bump? Or is the driver that we found in a cancer from, say, that intergen list, is it down in the tail? And therefore we have like a functional test, if you like, to evaluate those drivers. Because we can say, is our driver associated with evidence of selection, yes or no? And so we effectively, using this framework, we have a in vivo functional test of driverness on a tumor by tumor basis. So that's the big idea. So I hope that makes sense. The problem is that most of the time it doesn't work. And it doesn't work partly, I think, also implicit with the question before about what's your alternate hypothesis. So the assumption, of course, in our mathematical models that I've showed you to date is that there's no space. But of course, tumors are real spatial things, and spatial sampling really biases what you can see. So a few years ago, we developed in our wet lab a novel method to stain for mutations. It's based on RNA, and you can have probes for specific base pairs in the RNA. And it's a bit like doing immunohistochemistry, but you're doing it for the RNA mutations. And so we stained a whole load of colorectal cancers because actually these driver alterations, well, mutations in these genes are rarely subclonal in colorectal cancer, but we found a few where they were. And these are the data that we got. So these are all colorectal cancers where it's unshaded. There is cancer material, but it's not the tumor cells. This is the stroma. Where it's yellow, it's wild type for the mutation at the top, and where it's red, it's mutant for the mutation at the top. And so you can see really nicely in these three cases that there's complex spatial structures in cancers. And then you can imagine, like, if you were to sample from these tumors, you can get a very distorted idea of what the evolutionary dynamics are from a particular sample. You know, for example, if we sample the middle one here, you could sample over in this yellow patch and say, well, there's no interesting selection in this cancer, but it's clearly if you sample here where the RAS mutant is, you might call selection. And so you both risk missing a selected subclone, but also your measurement of the size and the number of mutations in a clone is also sample dependent. And so ideally, you want to have a framework where you can use the discovery power, if you like, of being able to sequence a bunch of stuff through multi-region sequencing and integrate it into these mathematical frameworks. And so that's what we've tried to do more recently. Excuse me. And so what we've been doing is taking those stochastic branching process models and sticking them into 2D or sometimes even into 3D. Sorry, excuse me. You can just appreciate the really beautiful pictures in a second. All right. So we take the same framework as before. We take neutral as our kind of null hypothesis. So what do the data look like if there's no selection, if nothing interesting is going on? And then on the right, we have cases where we simulate selection. So the top panel here, so X and Y are space in our 2D tumors. The color on the top panel is the subclone identity. So where it's red, that subclone has an advantage, and you can see it's growing more, bulges out here. And on the bottom, it's the same tumor, but now rather than coloring by subclone identity, we color, if you like, by lineage identity. So we can just do some projection of the mutations in a lineage into color space, and you can get these plots. So now similarity of color indicates shared ancestry. And so you can see the clonal structures that exist in the simulations. So then what we can do, of course, when we have these models is we can simulate the process of spatial sampling and mimic what we might do in the lab. And then, of course, we can also simulate sequencing data on top of these biopsies using some of the tricks that Simon introduced me 2 years ago. And then we can produce synthetic phylogenies from our simulations. And so when you do that, you can see that the shape of the phylogeny depends on the evolutionary dynamics in the tumor. So on the left are 2 phylogenies from a neutral tumor, and on the right 2 from a tumor that's got strong subclonal selection. And here the one on the left of each pair is when we take just 10 samples from the tumor, and the one on the right of each pair is when we take 400 samples from the tumor. So that might be analogous to single cell DNA sequencing, say. And so you can see that there's a distinctive difference in the tree shape. Essentially, when there's selection, you get sticky out branches. And if you sample evenly, those branches are much more bushy under selection. And so, you know, you get it kind of makes sense, right? If a clone's growing fast, then it pulls out all the passenger mutations to mark the clone, just as it does in the dimensionless case before. And then you oversample the clone because it's more common in the tumor than it would be neutrally. So you can see that there's characteristic patterning in these trees when you have selection. And so sort of armed with this knowledge, we can go in then and then interrogate our phylogenetic trees that we derive from multi-region sequencing and assess whether or not, and because we've annotated drivers on these trees, we can then say, is the driver on our tree actually associated with a clade, which we can see is under selection in this simulation framework. So hopefully it still makes sense. So with my close colleague for many years, who I met through knowing Simon, in fact, Andreas Otteriva, who was at the ICR for a while, but now has moved to head a computational biology center in Human Technopole Milan. Andrea and I had a grant from the Wellcome Trust in the UK, which was called EPIC, stands for Evolutionary Predictions in Colorectal Cancer. And that funded us to, sorry, again, slightly gory picture, funded us to generate this really amazing data set. So what we did is take a number of primary colorectal cancers, and we sampled them in a very ordered fashion. So we sampled around the clock, taking 4 large samples, which we then subdivided each sample into a number of individual glands. So you may know that the normal colon is organized into colon crypts, and those crypt structures persist through cancer evolution most of the time. So even late stage cancers still have these glandular structures. We think each gland is maintained by a small number of cells in the long term, so their progeny are lost from the crypts. And so effectively each gland is like a mini PCR machine that amplifies the DNA of those few long-lived cells in the gland. And so what we did is pick out individual glands and then treated them like they were a clone. And effectively then we could do clone by clone sequencing by sequencing each individual gland. And because there's maybe 10,000 cells in each gland, we're able to do very high quality multi-omics. So on each individual gland, we did deep whole genome sequencing to about 50x. We did attack sequencing to look at the patterns of open and closed chromatin across the genome, and also full transcript RNA sequencing on the exact same samples. And we generated this huge set of data with pretty good overlap between the omics. We did much more attack-seq because it was cheap, and the data were quite unexplored as well. And so here we have spatially resolved multi-omic data from a number of colorectal cancers. And so focusing on the genetic data, then we can, using the standard methods, we can make phylogenetic trees from each of the cases that we have the data from. So here's our forest of trees, just because I used the color code throughout the rest of the talk. So here's the depiction of the tumor, and the colors represent where the samples came from in space. So within region A, we took a bunch of samples, and the same within region B and so on, and those colors follow through the talk. All right, so I can see some of you looking at the trees, perhaps trying to spot which ones have that pattern of selection, have sticky out branches. You've probably spotted some. I think this one is the clearest one. And this clade here actually has you may not be able to read it, but this is a well-known KRAS mutation. And so you would suspect this clade's under selection. If you look at the rest of the tree, I think it's quite hard to see other clear examples. There probably are some in there, and if you look hard enough, you may be spotting some, but they're not very clear. And so I think my first takeaway when I looked at the data was that even when you have this you'd expect high-powered way to see selection, you don't see it very often in colorectal cancer. They all look pretty neutral-like. All right, but of course, we're quantitative, and I learned about ABC. And so we wanted to be able to do something a bit better than just eyeballing the trees and asking which ones have roughly the right shape. And so what we did is try and we used ABC to fit synthetic phylogenetic trees derived from our simulations, where we could mimic the actual spatial sampling that we had from patients by sampling in the same locations in our tumor, and fit our simulated trees to the ones we'd actually observed in individual patients. So we calculate a distance metric across the comparing our tree to the simulated data, and then we use ABC to fit the parameters of our spatial model, which are parameters like how fast is the subclone growing, how pushy is it to squeeze into new space, what's the rule for moving the other cells out the way, obviously what's the background birth and death rate, those kinds of things, and we fit the trees. And importantly, relating back to the question earlier, this lets us do model selection, where we can compare whether or not the data are best supported by a model where everything is neutral, or whether there's one subclone or two subclones or even more subclones. And we can ask what's the most likely explanation. And then these are some illustrations of the posterior distributions after doing that. I mean, you can't really read the axes, I appreciate, but you'll see they're fairly pointy distributions, so we can get some reasonable confidence in our answers. And if we look at the posterior predictive distributions, they don't look too bad either. And it's kind of amazing that it works so well, that the prediction's not so bad, because we used 2D simulations most of the time. And you'd imagine that 3D really messes things up, but it seemed not to. And it also didn't matter so much which statistics we used to compare the trees either. Okay, so this means then we can do this inference, and on a case-by-case basis, we can have some quantitative prediction about whether or not that phylogenetic tree shows evidence of selection, and say something quantitative about what the selective advantage is. And so when we did that, this is a summary across the 26 cases where we had sufficient data to make a good tree and do the inference. And you can see that only in a small minority of cases were we very confident from the posterior distributions that there was subclonal selection going on there. The majority were neutral, and then there was a set where we couldn't really decide. So there was some evidence for selection, but it wasn't particularly greater than the neutral evidence for neutral, or vice versa. So we weren't sure what was going on. And so the summary would be that there's only strong evidence of subclonal selection in a small number of cases. In fact, it was 6 of the 26. And then to have a kind of orthogonal test, we went back to trying to use that intergen-like approach, where we could take the average sequence information across our whole cohort. So we divided the cohort then into cases where there was selection and ones where we were confident there wasn't selection. And then we looked at the NDS, whether or not there were more non-synonymous mutations than not in the cases where we call selection in driver genes, compared to the cases where we said the tumor was neutral. And you can see that there was an enrichment for higher DNDS for missense mutations in cases where we call selection that wasn't there in those putative driver genes in cases where we'd called it neutral by this other method. And so it kind of worked, and it's verified by this orthogonal method. So when we've got this, then that lets us go back, as we've been talking about throughout, and make predictions about the driver status of individual genes in individual cancers. And so this was my poster child case that I showed you earlier. This is the one with this really sticky out long branch, looks exactly like those simulations I showed you earlier on. And this one has the KRAS G12C mutation on this branch. So we would conclude this KRAS mutation really is operating as a driver in that particular cancer. Whereas in these other ones that I showed you earlier on, these ones are both called neutral. And so this PI3 kinase mutation, and PI3 kinase is an interesting gene. Some people think it's really important for cancer biology and others are less sure. But in this particular case, there's this subclonal mutation in one of the known hotspots, but it doesn't seem to be predicted to be a driver by our simulations at all. And then here, there's lots of interesting mutations going on. None of them are predicted to be drivers, but I think this illustrates really nicely some of the caveats of the approach. But I think it's the caveats are, it's perhaps the wrong word to use, but what the simulation says is that at the point in time we sample the tumor, there's not good evidence of strong subclinical selection in this case. But you can see, for example, there's this FBXW7 mutation. It's a truncating mutation, but it's only present in one sample. So it's not to say that in the future, this clone wouldn't have taken over the cancer if it was left in the body. But at the point in time when we sample, that mutation is not doing anything. But I think to call it a caveat is maybe being harsh to myself, because if you were thinking about, say you had a drug for that mutation, you probably wouldn't want to use it in this case, given you'd expect that drug to have side effects, because at the moment that mutation really isn't doing anything for the tumor. But if the tumor was left in, then you might want to do it. So you can see how time affects driver status too. So it may be that that gene is a driver but just showed up a bit late. So why is there a mismatch between our understanding at the cohort level of what a driver is and what we see in a tumor? I think there's lots of explanations, but one might be that there's selection to particular microenvironment, and the microenvironment might be temporally changing. And so it may well be that those mutations have shown up, but they haven't experienced the right environment yet to drive a clonal expansion. They may become drivers, say, if the patient had a particular treatment down the line too. So selection is always context dependent, of course. All right. How am I doing for time? Fine, I think. So if we put all the data together and we ask how often do we find Ryan Gosling, so in the 6 cases where there was a known subclonal expansion, 4 of them had a Ryan Gosling, had a driver mutation that we had thought was there and was really there. So that meant that 2 of our 6 cases where there was subclonal selection didn't have a known driver. So there is still more to be learned, I think, about which genes or sets of genes together drive cancer evolution. And then importantly, if we think about then what the sensitivity of that antigen list is like for calling driverness, and you plug these numbers in, you get that the sensitivity of the list, if there is a driver there, how often is it contained on that list? It's about 2/3. And then the converse, how often is there someone masquerading as Ryan Gosling? In the UK, this joke works really well because that's Harry Kane, who's captain of the English soccer team. And he does look a bit like Ryan Gosling. And also, he missed a penalty in the Euro final, so everyone knows him very well now. And it's fine to tease him. Anyway, so how often is there someone masquerading as Harry Kane? How often is there a driver in a putative driver in a cancer that's not really a driver? And it's a lot of the time. So in 15 of the cases, 11 of them have mutation in a driver gene that isn't actually functioning as a driver. And so if we equate that to the specificity of that driver list, it's really quite low. It's only about 1/3. And so that's interesting. So it might be that these mismatches between drivers is quite common. Drivers in the microenvironment is quite common. So probably 5 minutes left, is that? Yeah.
More if you want it.
Okay.
Sort of wear you out. Perform tomorrow as well.
Oh, that's all right. So I thought I'd talk a little bit about the epigenetics because it's quite unexplored and I think it's quite interesting. So this bit is less mathsy, but I think the data are quite cool. So we had in the EPIC study, we sequenced normal intestine tissue next to our cancer, as well as doing that multi-region sequencing in the cancer. And we did that with the TAC-seq, so we can look at the pattern of chromatin organization in the cancer compared to the normal, and therefore work out what's a somatic change in the cancer. And because it's multi-region data, we can use an evolutionary argument to say if a change is clonal, an epigenetic change is clonal in the cancer, then it probably happened early. So it's probably in that first cancer cell in the cancer. And so what you're seeing here is data at one locus from one cancer. The orange line is the chromatin accessibility in normal. So you can see it was very closed in normal for this gene, JAK3, potential oncogene. Whereas it was open, we got reads here. The y-axis is the normalized read coverage at that location in all of the cancer regions. So it looked like this gene became clonally accessible early in cancer evolution. And actually, we saw this kind of pattern of somatic chromatin accessibility alterations or scars for a number of genes across the genome. And it made us wonder if just changing chromatin patterns was sufficient to sort of turn oncogenic genes on. So it may well be that epigenetic changes themselves could function as oncogenes. Obviously, this isn't direct evidence of it, but I think it is, somewhat persuasive. So if you put it all together, here's genes that had recurrent changes in promoter accessibility, red becoming open or blue becoming closed for promoters or enhancers across our cohort. And if you can read the gene list, you'll see that there are a number of genes on that list which are known to be oncogenes in various types of cancer. And importantly, here in our colorectal cancer, we could see changes in accessibility of those genes that can putatively control their expression that occurred without there being a mutation. And so you could get these chromatin changes affecting tumor biology without there being genetic mutation. So it may well be that scars themselves could be functioning as drivers sometimes. We also saw global rewiring of the transcriptome. So if we average the accessibility of transcription factor binding sites across the whole genome and ask which transcription factor binding sites tend to become more closed or which other ones tend to become more open, and then do a pathway analysis on what that would mean for gene expression programs across the whole genome. And what's enriched there is regulation of interferon gamma signaling and also some kind of regulation of like stem-like behavior. And so it seems like there are common recurrent changes in the global rewiring of the epigenome that associate with two phenotypes which are putatively important for cancer. You can't really read this graph, but it looks pretty. Okay, so it may well be a role for the epigenome in cancer evolution. And so going back more to this modeling framework, because we have the genetics and we have the models to say what's going on in the genetics, we can then use the other omics and overlay that on top and say, well, what's the phenotype of a selected clone? So this is one of the 4 cases where there is a selected subclone, and here it has an RNF43 truncating mutation in this subclone. And you can see there's this slightly sticky out branch of the purple clone here. And then we can say, well, what's the gene expression of because we've got matched RNA-seq, remember, of the samples within that clone compared to the rest of the tumor. And we can get an in vivo readout in a patient's tumor about what the phenotype of that selected subclone is. And you see a whole bunch of changes that if you do pathway analysis, there's various metabolic changes going on there within that subclone. So you'll remember as well that there were 2 of 6 cases where we called selection, but we couldn't find a driver. And so we can use the same approach to say, to try and discover epigenetic changes that might be functioning as drivers. So again, there's selection here. It's actually this subclone that's selected, even though this one looks a bit more sticky out, because you can see there's 3 different colors in this subclone. So there's this massive clone that's spread across 3/4 of the tumor with virtually no new mutations acquired in the interim. So it's obviously growing very, very fast, which is why selection is called here. If we look in the RNA-seq, there are a bunch of changes in the subclone compared to the rest of the tumor that correlate with there being epithelial to mesenchymal transition in the subclone. And then if we look in the ATAC-seq, again, there's a whole bunch of chromatin accessibility differences between the tumor regions. I've just cherry picked some genes here because I have no other way of saying what's most important. But one of the most stark changes in the subclone to the rest of the tumor is this loss of accessibility at this locus, which is some gene that regulates p53 activity. So I've pulled it from thin air, but it's suggestive. But you can imagine that this kind of approach might give you a route to interrogate functional changes in the non-coding genome. I don't want to overrun my time, but I can talk very briefly about plasticity. So because we have the genetic history, and we also have the phenotype, the gene expression phenotype and chromatin accessibility of tumor cells, we can start to look at how phenotypes evolve and what their relationship is to underlying genetic change. And so to do that, we need to define, and we're really interested in plasticity. It's a hot topic in cancer, and so we really need to know what plasticity is. And so I think in the field of evolutionary biology, the classical definition of plasticity is a chameleon, where an individual, be it a cell or an animal, can change its phenotype within its lifetime. That's, I think, the right definition, but it's not something that's very useful for studying cancer because of this problem that we can't measure over time that I talked about before. And so we need to have a working, a more practical definition to interrogate plasticity. So our definition then is the heterogeneity of phenotype within a clone. And so here's all the stormtroopers are all the same, apart from this guy who's got a red thing on his shoulder. And so he's a standout within the clone. So he looks plastic compared to the rest of that clone. So this is the definition that we run with, and we can use it to interrogate the data. And so let's go through that one. So what we have is this setup where we have a phylogenetic tree on the left here, and we can interrogate the shape of the phylogenetic tree as I've described to understand selection, evolutionary forces. And then for each sample that we've taken, we also have its phenotype, at least its gene expression pattern. And we can ask, well, how often does that gene expression pattern correlate with the structure of the tree? So in this particular tree, this is the one with the KRAS mutation. These genes all have high expression within that subclone, and they correlate with the evolutionary history. And so they're associated with that expanding phenotype of the RAS clones, whereas these mutations in my cartoon have no correlation with the tree structure. And so we can say that these genes are not plastic because they follow the tree. Their expression is predicted by their position on the tree, whereas these genes are likely to be plastic or just noisy because they don't follow the tree. They don't have a signal which is explained by evolutionary history. There's some tools in phylogenetic in evolutionary biology which are well established, which are called phylogenetic signal, and there's various statistics you can compute about the correlation structure of genes over the tree. And one of the popular ones is this one called Pagel's lambda. So what we did is run Pagel's lambda on our data. We realized very quickly that our data was a bit underpowered here, so we didn't find very much. But I think that tells us that most of the time there isn't a very strong phylogenetic signal in cancers. So this is one of the best examples. Here we've plotted pathway activity of particular genes. So the color here is the activity of these pathways, which I'm sure you can't read, but these are the hallmark pathways. On the left is not a dendrogram, it's the phylogenetic tree from the whole genome sequencing for all these samples from a particular cancer. And what you can see if you eyeball it straight away is that there's these patches here and this sample here, which have very similar phenotypes, similar gene expression programs, but they have very different genetic histories. And so we're seeing a kind of convergent evolution of phenotype and a plasticity here that you can get to the same phenotype independently of the evolutionary history. So that I think that's nice evidence of phenotypic plasticity directly in vivo in these patient tumors. And then if we apply this Pagel-Lambda statistic, we see that only a very small number of genes, about 2%, have heritable patterns of gene expression, at least given the power that we have to see this in our data, which I emphasize is not very great. But that means there aren't strongly heritable patterns of gene expression very often in colorectal cancer. And we can also ask, well, how often do mutations correlate with the change in gene expression locally? And that's also quite rare. And so genuinely, most mutations in cancer really are passengers that don't do anything. Okay, so I'll stop there. So I've talked about how selection, how evolutionary dynamics shape the pattern of heterogeneity in tumors, and how we can use models to read that pattern. And that gives us, if you like, this dynamics from a snapshot. And then if we overlay other omics on top of these models and that genetic data, we can start to, I like to think of it as functionally probe what's going on in vivo in patient tumors. And perhaps surprising finding is that many genes that we think are important, and certainly are important on average, but might not be important in the tumor in front of us. And that potentially has consequences for things like personalized medicine down the line. We also see commonplace, we also see evidence for plasticity being commonplace in colorectal cancers. And I think that's a really interesting thing to try and understand going forwards. So yeah, thanks a lot for listening. Lots of people have done this work. Principally, Jacob did all the gene expression and plasticity stuff. Timon, who was a PhD student principally with Andrea, did all of the modeling work that I talked about at the start. And some of the earlier modeling work without space was done by a number of other people, including Mark Williams, who is now at MSK here. And it's been a big collaboration with Andrea for a large number of years. And principally the work today is funded by Wellcome. So thanks very much for listening. This is what we look like. Yeah. That's Darwin's house as well, by the way. It's his famous balcony, and there's that picture of him next to the pillar, which is this pillar here. There's only a few actual pictures of Darwin. It's in southeast London. If anyone's ever in London and wants to visit the house of him, it's a National Trust place. You can go down.
So, Trevor, maybe you could address questions on the chat first.
Sure. So there's one from David Axelrod, who asks, is there an implication that it would not be useful to target drivers for chemotherapy? I think, David, that I like to see it that this kind of method lets you make decisions about a particular tumor on a tumor by tumor basis. So if you had a drug for a particular gene or a particular mutation, you could use this kind of methodology to assess whether or not that gene or that mutation was important in the tumor that's in front of you, and use the drug accordingly. So I hope it can aid decision making eventually, but of course it's just an idea right now. It's just hypothetical. I think that's it.
We've got a question back there. I think we mind repeating it.
Sure.
I have a question about the definition. I'm great.
Thank you.
For selection to happen, the subclone will have to be bigger than the dominant clone of the tumor in the first place.
Yes.
So without, I assume those are tumor-naive cases. Without this external stimuli, you not see the selection. So the fittest clone is already the one that over to call some first place. Is that fair to say?
Yeah, that's fair to say. So the for those listening externally was comment that the tumors we've looked at are untreated, and therefore I think, to paraphrase, that selection might have already done its job and assorted the clones in the tumor to make the fittest one dominant. And so perhaps it's not surprising that we don't see selection very often. I think that's a fair comment.
And did you try that approach where so you can also kind of find the clonal mutation based on what prior or after well,
yes.
Like, do you see any patterns in this? Or
So that isn't an assessment of selection, though, is it? That's just an assessment of time. Unless you think that amplification of a mutation, say, is evidence of it being selected, which it might be, but it's not a direct functional evidence of it being selected. So those of you listening remotely, the question was, did we time mutations using the fact that copy gains are timable by the fact they increase the allelic frequency of mutations that occurred before the gain? So I think timing and selection are different things, and we shouldn't convolute them.
So a lot of the theoretical results depend heavily on the assumptions in the model. So for example, the VAF might depend on whether the growth is more exponential or statistical or constant or something. And then I think there were some recent papers showing that the constraint on the spatial modeling might have a lot of effects on the pattern that you see. Right? So how much does that affect?
Yeah. So the question was, how do the assumptions of the model, particularly around space and spatial sampling, affect the outcome? So I can answer in two parts. So clearly space is important, which is why we model in space. One of the things that we're able to infer from the shape of the tree is the likely mode of growth, be it boundary driven or purely exponential. They shape the tree in different ways. In fact, there was a really nice paper that came out recently. That right now the name slips my mind, but from a new group in Washington that really nicely interrogates the shape of the tree given the dynamics of the population growth. So yeah, you can infer some things about that kind of spatial structure. And then in our earlier work where we didn't think about space, I think ironically one of the things that saves us from not suffering all the consequences of space is that data are not very good. And the reason I think, and that's obviously ironic because it becomes more of an issue when you've got better data, whereas you think with better data things should be easier. But I think with worse data, if you think about it, like very good genome sequencing on bulk is say 100x, or I don't know, if you're made of money, it might be 500x whole genome. But still the number of alleles that you're sampling compared to the number of alleles that actually exist in the tumor is a tiny, tiny, tiny fraction. And so that means that you only see resolving the data, the very early events in tumor evolution, where it's probably not unreasonable to think about exponential growth happening, and that spatial effects are unlikely to be that important because the tumor is so small. And so I think, ironically there we're saved by the bad data. And I'd expect that theory to look increasingly worse against the data as the data improves in the years to come.
So another question I have is, how long did it take for you to make one of those 2D/3D simulations?
Wow, I think Timon spent his whole PhD doing them, so good 4 years to get good answers out, and there was lots of back and forth. The code is available though, so you can take all of his fantastic hard work and just run with it.
So how long does it take to make 1 million simulations?
Oh, I don't know off the top of my head, but it's not that bad. So we do various tricks, which Simon had introduced me to a long time ago, about only simulating the mutations in the cells that you need to simulate them in, by having a kind of, three, well, I guess it's not 3-pass, but a 2-pass approach. So you can simulate the branching and then throw away the rest of the tree that you haven't sampled and just simulate mutations over the bit of the tree that you retain, that kind of thing.